Hello friends, this is Manisha from Food Tech Network. Today we are going to discuss about some questions regarding food processing technology. Before we start our session, I would like to request you to all please do subscribe our channel Food Tech Network. Okay, so let's get one. Water activity can act as a dash. Option A, an intrinsic factor. Option B, processing factor. Option C, an intrinsic factor determines the proliferation of microbes. And option D, all of the option D, that is, water activity can act as an intrinsic factor, a processing factor, as well as intrinsic factor that determine the likelihood of microbial proliferation. It also helps to determine the shelf life of food product, and it affects the growth and survival of microorganisms in food. Understanding water activity is essential for proper food microbiology. Question number 2. Suspected colonies of Staphylococcus aureus, when grown on a Bayard Parker medium, shall show dash. Option A, protease activity. Option B, catalase activity. Option C, coagulase activity. And option D, none of the above. So the right C, that is coagulase activity. Staphylococcus aureus is a type of bacteria that produces coagulase enzyme, which also helps them to attach and colonize the host tissues. When grown on a Bayard Parker medium, colonies of Staphylococcus aureus will show a coagulase activity. An undesirable change in a food that makes it unfit for human consumption is referred to as a dash. Option A, food decay. Option B, food spoilage. Option C, food loss. And option D, all of the above. So the right answer B, that is food spoilage. Basically, food spoilage is caused by bacteria, molds, and other microorganisms that contaminate the food and causes it to become unsafe for consumption. It can lead to off flavor, discoloration, texture changes in the food. As well as preventing food spoilage requires the proper storage of foods, careful handling, preparation, and paying attention to the expiration date. Which of the following statements are true regarding botulinum toxin? Option A, botulinum toxin is a neurotoxin. Option B, it is a water soluble exotoxin. Option C, it is produced by Clostridium botulinum, a gram positive anaerobic bacteria. And option 4, that is option D, all of the option D. Basically, all the three statements are factually regarding the botulinum toxins. Botulinum toxin is indeed a neurotoxin because it acts by blocking the release of acetylcholine, a neurotransmitter responsible for transmitting signals between nerve cells and leading to muscle paralysis. Option B, that is botulinum toxin is a water soluble exotoxin because exotoxins are the toxic substance that produced and released by a bacteria which can be dissolved in a water. So the third option is Clostridium botulinum, a gram positive anaerobic bacteria produces a botulinum toxin. One of the most potent toxin known to human is a botulinum toxin, which is produced by Clostridium botulinum bacteria. Clostridium perfringens poison is N dash. Option A, exotoxin. Option B, enterotoxin produced during sporulation. Option C, endotoxin. And option D, enterotoxin produced during the vegetative phase. B, that is Clostridium perfringens makes an enterotoxin during its sporulation phase, resulting in a food poisoning and gastrointestinal illness if food is not properly or correctly cooked or stored at required temperature. Aerobic colony count, that is ACC, is referred to as a dash. Option A, total viable count. Option B, aerobic plate count. Option C, standard plate count. And option D, all of the above. Option D, that is all of the above. Basically, TVC, APC and SPC is also referred as a ACC. Apart from that, SEC is a method used to measure the bacteria in food. It uses culture techniques such as agar plate to determine the number of viable bacteria cells per sample. Aflatoxin is produced by dash. Option A, Aspergillus species. Option B, Salmonella species. Option C, Fusarium species. And option D, Streptococcal species. Option A, that is Aspergillus species. Aflatoxin is produced by a fungi belonging to the species of Aspergillus, and it can cause a severe health problem when consumed through a food. It is also important to note that the other bacteria such as Salmonella, Fusarium as well as Streptococcal may contaminate the food product. However, they will not produce any kind of aflatoxins. Find the incorrectly matched pair. Option A, Serratia, a drug addiction. Option B, Spirulina, single cell protein. Option C, Rhizobium, a biofertilizer. Option C, Streptomyces, an antibiotic. Option A, that is Serratia, a drug addiction. Basically, Serratia is a type of bacterium and it is often used to produce antibiotics and biofertilizer. On the other hand, addiction is not associated with this type of bacteria. Enumeration of microorganism refers to dash. Option A, non-selective plating depending on the taste. Option B, either spiral plating, pour plate or spread plate of a food suspension onto a suitable selective agar. Option C, either A or B, and option D, none of the above. Option C, that is A and B, both the sentences are correct because it allows for the isolation and enumeration of the microorganism present in the sample. Botulism prevention involves option A, proper heat sterilization before food canning. Option B, addition of chemical preservative. Option C, proper low temperature treatment before food canning. And option D, all of the above. Option D, because proper heat sterilization before food canning is essential to destroy the spores Clostridium botulinum, the bacteria which is responsible for producing toxins. Second is addition of chemical preservatives such as acids or antimicrobial agents which can inhibit the growth and activity of Clostridium botulinum and other bacteria. And this preservative create an unfavorable environment for bacterial growth and toxin production in food products, contributing to botulinum prevention. The third option, which is low temperature treatment, is essential and it is also known as a refrigeration or chilling. It also inhibits the growth of Clostridium botulinum and other bacteria by storing perishable food product at a low temperature, The bacteria's growth and toxin production can be slowed down or prevented, and it can ultimately reduce the risk of botulism toxins. Thermal destruction of microorganism follows the kinetics of option A zero order, option B first order, option C second order, and option D, that is fractional order. Option B, that is first order. Food microbiology study microorganism present in or it can spoil our food. Thermal destructions follows the kinetics of first order, meaning that the particular amount of heat energy is needed for each microorganism to be destroyed. And this means that as the temperature increases, the destruction rate of the microorganism will increases simultaneously. Niacin are produced by dash Aspergillus niger, option B Acetobacter aceti, option C Lactobacillus lactis, and option D Clostridium perfringens. C, that is Lactobacillus lactis. Niacin is a bacteriocin, a type of antimicrobial peptide produced by a certain strain of Lactobacillus lactis, and it is also naturally occurring antimicrobial compound widely used for a food preservative to inhibit the growth of various foodborne pathogens as well as strains of gram-positive bacteria. A plating medium consisting of dash is commonly used for bacterial enumeration in food. Option A peptone, glucose, sodium chloride, agar, and distilled water. Option B yeast extract, glucose, sodium chloride, agar, distilled water. Option C peptone, yeast extract, glucose, sodium chloride, and distilled water. Option D peptone, yeast and extract, glucose, sodium chloride, agar, and distilled water. D. This combination of ingredients provides the essential nutrient needed for a bacterial growth, and it also contains agar, which act as a gelling agent, allowing the bacteria to be plated consistently. The top contaminant in the canning industry is dash. Option A lactobacillus, option B clostridium botulinum, option C bacillus subtilis, option C trichoderma polysporum. Answer is option B, that is Clostridium botulinum. It is an anaerobic bacterium that can cause a potentially deadly food poisoning known as a botulism, and it is also found in soil and can contaminate the canned product, particularly those with a low acidity. Inadequate heat processing or improper storage can lead to the growth of this pathogen, and it should be avoided at all cost. Question number 15. Trichoderma hertzanium is helpful for dash. Option A. Bioremediation meditation of contaminated soil. Option B. Gene transfer in higher plants. Option C. Reclamation of wasteland. And option D. Control of soil-borne plant pathogen by biologic. Option D. It is a fungus species known for its beneficial properties in agriculture and horticulture, and it has been extensively studied and utilized for its ability to act as a biological control agent. Soil-borne plant pathogen. Trichoderma can also colonize the root system of plants and suppress the growth of various harmful fungi, including plant pathogen. It does so by competing for resources, producing antifungal compounds, and stimulating the plant's natural defense mechanism. 16. How is the host plant's resistance to a disease assessed? Option A by examining the leaves, option B researching the seed, option C analyzing the genetic makeup of the plant, and option D by cutting the stem. C. The genetic makeup of the host plant affect the resistance of the host plant, which is capacity to stop the pathogen from producing illness. And the causal organism and the transmission method must be understood prior to the breeding. Examine the leaves or a seed can help in determining the presence of any foreign pathogen or insect, but it cannot show if the plant is resistant to that pathogen or not. Such information is generally stored in the genes, which are expressed in the form of toxic proteins. What is the following does not cause a crop losses? Option A fungi, option B bacteria, option C viruses, and option D, that is earthworm. Numerous bacterial, viral, fungal disease, especially in the tropical climate, have an impact on the production of farmed crop species. Crop losses can frequently be substantial, reaching 20 to 30 or even being completely lost. Earthworms do not cause a crop losses but rather create a nutrient for the plant that help in increasing the crop yield and fruits and vegetable quality, and also reduce other harmful pathogens in the soil. They convert the organic debris into compost, which can be applied directly on the crop field to provide a good quality nutrient to the plants. Which of the following term is used to describe the organisms that are under the attack of pathogen? Option A crop, option B variety, option C host, and option D susceptible host. So the right answer is option D. It is a susceptible host. When an organism is attacked and injured by a disease yet lacks immunity to it, it is referred to as a susceptible host. Hosts are vulnerable individuals within which a pathogen causes illnesses. The intermediate host, also known as a vector, is typically an arthropod or mollusc, where the definitive host, which harbors the pathogen's mature form, is frequently a vertebrate. The Zika virus, Lyme disease causing a bacteria, and malaria causing protozoa all exhibit this alternative between the vertebrate and intervertebrate host. Number 19. Which of the following describe a wheat crop variety? Option A Pusa Swarni, option B Pusa Shubra, option C Himgiri, and option D Pusa Komal. Answer is option C. Wheat is grown in a cultivar called Himgiri, and brassica, cauliflower, cowpea respectively comes in the variety under Pusa Swarni and Pusa Shubra and Pusa Komal respectively. So the wheat cultivar is called Himgiri. The two most popular Himgiri variants is HS375 and HS375 on irrigating rain-filled land. HS375 is generated, while HS375 is produced at very high altitude, that is in the northern part of Himalayas and Garhwal regions, and it is the medium fertility and late sown hybrid of the Himgiri plant. Himgiri is resistant to heel burnt and leaf and stripe rust diseases. What benefit do plants that are resistant to disease provide to the production of food? Lessen reliance on fertilizer, option B lessen reliance on fungicide, option C lessen reliance on water, and option D lessen reliance on air. So the right B. Crop losses lead to the breeding and development of disease resistant cultivar, which boost the food output. Additionally, it lessens the need for fungicide and bactericide. This fungicide and insecticide are chemical used to kill the disease causing organism. However, when a plant is made disease resistant, they naturally develop a resistance to these pathogens and they need to reply This chemicals is greatly reduced. 21. Which of the following is not a necessary stage in the breeding process for disease resistant crops? Option A, crop harvesting. Option B, germplasm screening. Option C, hybridization of selected parents. And option D, selection and assessment of hybrids. Option A. The numerous sequential stages involved in breeding disease resistance crop include screening germplasm for a source of resistance, hybridizing, choosing parents, choosing and assessing the hybrids, testing and releasing the new varieties. Crop harvesting is not necessarily a part of the plant breeding process but comes later during the commercial growth for a sale and purchase, and the crop will harvested once the breeding process is completed successfully and it has been determined that the breeding is successful. 22. How do mutation occurs? Option A, change in DNA. Option B, change in the organism's overall genetic makeup. Option C, change in RNA. And option D, change in the base sequence of genes. D, that is change in base sequence of gene. The mutation is the process by which genetic variants are formed by changes in the sequence inside the genes, resulting in the production of a new character and characteristics not present in the parental type. The entire DNA or genetic makeup of an organism usually does not change because of mutation. Amputation may be positive if the new character benefits the organism and may be negative if the new character caused harm to the 23. Which of the following crops has chili mosaic virus resistance? Option A, cowpea. Option B, cauliflower. Option C, chili. And option D, brassicas. Option C, chili crop is resistant to leaf curl diseases, tobacco mosaic virus and chili mosaic virus. The bacterial blight is resistant to cowpea and black rot and curl blight black rot diseases are not harmful to cauliflowers, as well as white rust sickness is not contagious to brassica. 24. Bindi, which is resistant to yellow mosaic virus, has given a rise to a novel variety known as dash. Option A, Pusa Sadabahar. Option B, Parbhani Kranti. Option C, Himgiri. And option D, Pusa. There is option B, that is Parbhani Kranti. Bindi has developed a novel variety known as Parbhani Kranti as a result of receiving a resistance to the yellow mosaic virus. Sexual hybridization between the target and the source plant followed by a selection results in the transfer of resistance of genes. 5. Which of the following is employed to cause a plant mutation? Option A, radiation. Option B, water. Option C, virus. And option D, leaves. So the A. As using a chemical or a radiation, it is possible to cause a mutation in a plant's intentionally. Plants with the desired characteristics may then be chosen and used as a breeding source. Mutation breeding is the term used to describe this process. One of the fundamental principles of evolutionary biology is mutation. Because of damage or a copying error, when an organism's DNA mutate, it transmits the mutation to its progeny. The offspring are then either better or worse prepared to live and procreate. Researchers have also presumed, at least until recently, that such mutations are random and equally likely to occur along with a specific DNA fragment. 26. Which of the following does not constitute a improvement to the food production? Option A, agroforestry. Option B, plant breeding. Option C, tissue culture. And option D, deforestation. Option D, that is deforestation. The need to increase food production is urgent given the world's rising population. The cultivation of plants and the care of animal both play a significant part in raising food output. The development of new methods, including tissue culture, embryo transfer technology, will be essential to rising food production level. Deforestation involves cutting down trees, which does not enhance a food production but increases soil erosion and floods, and also damage the natural forest ecosystem, which promotes the growth of plant. Who is created with who is credited with starting the white revolution in India? Option A, Charles Darwin. Option B, M.S. Swaminathan. Option C, Herbert Boyer. And option D, Verghese Kurian. Option D, Verghese Kurian was the father of the white movement in India, which is why the answer D is correct. Milk production and the white revolutions are related. The National Dairy Research Institute's Operation Flood and White Revolutions are responsible for the increase in milk output in India. Which of the following procedure receives top priority in the administration of dairy farm? Option A, a rise in milk production and quality. Option B, rise in cattle size. Option C, rise in cow numbers. And option D, rise in buffalo numbers. So the A, that is rise in milk production and quality. A dairy farm is an enclosure where the dairy animals are kept and bred to produce milk. In dairy farm management, we work with methods and procedures that boost the productions as well as raise the milk quality. The dairy farm management practices should be such that the milk quality and quantity should be increased without any need to increase the number of cows or buffaloes. Nice. To maximize the yielding potential, which of the following must be done? Option A, cow should be left alone. Option B, cow should be well cared for. Option C, cow should be fed every other day. And option D, cow should have limitless access to fodders. B, that is, cow should be well cared. In order to yield more, the cattle must be adequately cared for. They must be housed properly, provided with enough water, and kept disease free. Cattle should be fed in a scientific way, paying a close attention to the kind and quantity of forage. So, last number 13. What breeding technique is ideal for animal whose output is below average? Option A, interceptic hybridization. Option B, outcrossing. Option C, inbreeding. Option D, outbreeding. So the right B, that is outcrossing. Outcrossing refers to the practice of marrying animals of the same breed who do not have any common ancestors on either side of their lineage. So, called outcross is the result of such a mating, and it is the ideal way of breeding for creatures whose development rates in a beef cattle or their productivity in producing milk are low average. Thank you for watching this video. Stay tuned, stay connect for more.